What is the most famous and deadliest of the puffer fishes?
Fugurubripes, the Japanese pufferfish, is a specialty in sushi restaurants. However, only specially trained chefs can safely prepare the fish for consumption. By minimizing the presence of tetrodotoxin, 1,000 times deadlier than cyanide.
What type of signals control cell reproduction?
Cell reproduction is controlled by both external and internal signals. External signals include the availability of nutrients and space for growth. An example of an internal signal is the rise and fall of protein levels at specific points of the cell cycle, which is maintained by checkpoints and feedback controls. Cyclins and cyclin-dependent kinases are protein regulators that activate cell cycles and stimulate the synthesis of proteins.
What is needed for DNA replication?
DNA replication is a complex process requiring more than a dozen enzymes, nucleotides, and energy. In eukaryotic cells there are multiple sites called origins of replication. At these sites, enzymes unwind the helix by breaking the double bonds between the nitrogen bases. Once the molecule is opened, separate strands are kept from rejoining by DNA-stabilizing proteins. DNA polymerase molecules read the sequences in the strands being copied and catalyze the addition of complementary bases to form new strands.
Who was Erasmus Darwin?
Erasmus Darwin, 1731–1802, was the grandfather of Charles Darwin, 1809–1882. Erasmus was a physician, inventor, and natural scientist who published a book on his ideas. Zoonomia, between 1794 and 1796. Zoonomia contained poetic couplets describing Erasmus Darwin's ideas about science and, in particular, the evolution of life. Erasmus Darwin's hypothesis was that all the animals on the planet had their origin in a vital spark that set in motion life as we know it. In his posthumously published book Temple of Nature, 1803, Erasmus Darwin further speculated on a theory of evolution that included basic ideas on the unity of organic life and the importance of both sexual selection and the struggle for existence in the evolutionary process. Years later, in writing a biography about his grandfather, Charles Darwin acknowledged that his grandfather's ideas had influenced his thinking.
How do bats hunt?
Bats hunt a variety of prey. Some bats are insectivores, capable of catching their prey on the wing. Vampire bats hunt for large, slow-moving organisms to use for sustenance. Frugivorous or fruit-eating bats are adapted to finding ripe fruit distributed throughout the forest, depending on the food type. Bats may rely on their sight and or echolocation to find their prey. In echolocation, bats emit ultrasonic sounds that bounce off the objects around them. These signals are fielded by the specialized folds of flesh on the face and around the ears, allowing the bat to judge the direction and distance of objects.
What is the standard procedure for differential centrifugation?
Differential centrifugation is a technique commonly used by biochemists. Tissue, such as liver, is homogenized at 32 degrees Fahrenheit 0 degrees Celsius. In a sucrose solution that contains both a buffer to stabilize the pH and a salt. The homogenate is then placed in a centrifuge and spun at a constant centrifugal force at a constant temperature. After a predetermined amount of time, a sediment forms at the bottom of the centrifuge tube and is covered by an overlying solution called the supernatant. The overlying solution is then placed in another centrifuge tube. The sediment after the first centrifugation is called the nuclear fractions and consists mainly of nuclei, which are the largest and densest organelles present in cells. The supernatant is centrifuged at a higher centrifugal force for a longer period of time. Another group of particles, usually the mitochondria, form the sediment. The process is repeated several times, each time at a higher centrifugal force for longer periods of time, until the sediment only contains enzymes and other substances not associated with any cell organelles.
What is evolution?
Although it was originally defined in the 19th century as descent with modification, evolution is currently described as the change in frequency of genetic traits, also known as the allelic frequency, within populations over time.
What is a telomere?
At the end of eukaryotic chromosomes there lies a unique structure known as a telomere. Experiments have determined that without telomeres, the chromosome structure may be compromised. The DNA of the chromosome tends to stick to other pieces of DNA, and enzymes, deoxyribonucleases, are more likely to degrade or digest the ends of the chromosomes. Human telomeres have specific repetitive DNA sequences, tag, that may be repeated from 250 to 1,500 times.
How did Malthus influence Darwin?
Both Darwin and Wallace read the work of Thomas Malthus, 1766 to 1834. Who in 1798 had published Essay on the Principle of Population. In it Malthus argued that the reproductive rate of humans grows geometrically, far outpacing the available resources. This means that individuals must compete for a share of the resources in order to survive in what has become known as the struggle for existence. Darwin and Wallace incorporated this idea as part of natural selection, that is, adaptations that made for a more successful competitor would be passed on to subsequent generations, leading to greater and greater efficiency.
How do plant cells store energy?
Plant cells store energy in complex carbohydrates such as starch, disaccharides, and lipids. These energy sources may be used to fuel cellular metabolism or to provide energy for the germination of seeds.
What is junk DNA?
The non-coding portions of the genome were formerly referred to as junk DNA. Some of this material turns out to play regulatory roles in DNA replication and transcription, and the rest may be an evolutionary holdover.
Why are insects often found in amber?
People have long been infatuated with amber, the fossilized form of ancient tree resin, a semi-precious stone used for jewelry and mosaics. Amber from the Dominican Republic contains an average of 1 insect in every 100 pieces. Some pieces of amber contain thousands of insects, both whole insects and insect fragments. These insects were probably crawling or lodged on the outside of a tree about 30 million years ago and became trapped by a glob of sticky tree resin, which continued to ooze around the animal matter and eventually fossilized. Scientists are able to study these insects, many of which are extinct but may turn out to be missing links to modern-day species.
How does an ultramicrotome differ from a standard microtome?
The ultramicrotome, developed after 1950 to prepare specimens for examination under the electron microscope, enables technicians to cut very thin sections, 50 to 150. Instead of embedding specimens in wax, small, 0.5 to 1.0 cubic mm. Biological specimens are embedded in very hard synthetic resins such as epoxy. In order to cut these very hard materials, special knives sharper than steel knives are used. The knives are either diamond knives or pieces of plate. Glass broken in a controlled way to produce a fine edge.
Do termites have any natural predators?
Birds, ants, spiders, lizards, and dragonflies have been seen preying on young winged termites when they emerge and fly from a home colony to establish new colonies. Termites are generally most vulnerable to predators when they emerge from their home colony. Chimpanzees are also known to use sticks as tools to forage for termites.
What are trichomes?
Trichomes are hair-like outgrowths of the plant epidermis. They are often found on stems, leaves. Woolly or fuzzy leaves are covered by trichomes, and reproductive organs.
What is the function of the Golgi apparatus?
The Golgi apparatus, frequently called the Golgi body, is a collection of flattened stacks of membranes. It serves as the packaging center for cell products. It collects materials at one place in the cell and packages them into vesicles for use elsewhere in the cell or transportation out of the cell.
What is the difference between a territory and a home range?
A territory is a defended area. It can be as small as the space around a female red-winged blackbird's nest or as large as the backyard that your dog defends. A home range, in contrast, is simply the area where an animal spends its time. Home ranges may be shared with conspecifics, members of the same species, and may overlap those of other species.
What are migratory beekeepers?
A migratory beekeeper is a person who transports his or her bee colonies to different areas to produce better honey or to collect fees for pollinating such crops as fruit trees, almonds, and alfalfa. The beekeepers frequently travel north in the spring and summer to pollinate crops and then back south in the fall and winter to maintain the colonies in the warmer southern weather. Approximately 1,000 migratory beekeepers operate in the United States, transporting approximately 2 million bee colonies a year.
Can animals suffer from psychological disorders?
Although animals are used as models for research into disorders like anxiety, depression, and even schizophrenia, these conditions have been induced in them either through surgery or behavioral treatments. Scientists are currently developing strains of mice and rats that are genetically predisposed toward these diseases in hopes of gaining a better understanding of symptoms and more effective treatments. As for animals in the wild, the answer lies in the still unresolved issues of whether animals experience emotions and whether they have consciousness or the awareness of self.
When was the term homeostasis first used?
Walter Bradford Cannon, 1871–1945, who elaborated on Claude Bernard's concept of the milieu intérieur, used the term homeostasis to describe the body's ability to maintain a relative constancy in its internal environment.
Are turtles endangered?
Worldwide turtle populations have declined due to several reasons, including habitat destruction, exploitation of species by humans for their eggs, leather, and meat, and their becoming accidentally caught in the nets of fishermen. In particular danger are sea turtles, such as Kemp's Ridley sea turtle, Lepidoscelis kempii, which is believed to have a population of only a few hundred. Other threatened species include the Central American river turtle, Dermatemis mawii, the green sea turtle, Chelonia midas, the leatherback sea turtle, Geochelone inophora, the desert tortoise, Gopherus agassizii, and the Galapagos tortoise, Geochelone elephantopus. Source: U.S.
What is the Red Queen hypothesis?
This hypothesis, also called the law of constant extinction, is named after the Red Queen in Lewis Carroll's Through the Looking Glass, who said, "Now here. You see, it takes all the running you can do to keep in the same place." The idea is that an evolutionary advance by one species represents a deterioration of the environment for all remaining species. This places pressure on those species to advance just to keep up.
What is the largest terrestrial mammal in North America?
The bison, bison, is the largest terrestrial mammal in North America. It weighs 3,100 pounds 1,406 kilograms and is 6 feet 1.8 meters high.
How many tentacles do the cephalopods have?
Octopods have 8 tentacles or arms, squids have 10 tentacles. And there are as many as 90 in the chambered nautilus.
What are sclerotia?
Sclerotia are the aggregate of hyphae enclosed in thick walls that form a protective covering when conditions temperature and water, are not conducive for growth. When conditions improve, the sclerotia germinate and produce stalks containing spore-bearing bodies. Ascospores are embedded in the tips of the stalks. When transported by wind, ascospores may land on grasses or grains, especially rye. The sclerotia continue to grow after they have landed on the host plants.
Do humans use pheromones?
Yes, human eggs release a chemical signal that allows them to communicate with sperm. Human females also respond to pheromones. In this case, the signals actually coordinate the menstrual cycle. In fact, 2 human pheromones have been identified. The first increases the likelihood of ovulation, while the second suppresses ovulation. For years, scientists debated the importance of pheromones to humans. But the close examination of the olfactory receptors and the vomeronasal organ in the nose has given researchers a mechanism by which humans may be able to recognize these compounds.
What is cytology?
Cytology is the study of cellular structure based on microscopic techniques. Cytology became a separate branch of biology in 1892, when the German embryologist Oskar Hertwig (1849–1922) proposed that organismic processes are reflections of cellular processes.
What are the functions of the central vacuole?
The central vacuole can encompass 80% or more of the cell. It is usually the largest compartment in a mature plant cell, and is surrounded by the tonoplast. The important functions it provides include storage, waste disposal, protection, and growth.
What is zero population growth?
Zero population growth, or ZPG, is the estimation of the birth rate necessary to maintain the size of the human population at its current level. As of now, the rate is estimated as 2.1, which means that each set of existing parents would need to have, on average, slightly more than 2 children during their lifetime. The extra 0.1 allows for infant mortality.
Why is it necessary to fix a biological specimen?
Fixing a biological specimen retains a reasonably good semblance of the object as it appeared when it was alive. It allows the scientists to observe details of the external and internal anatomy of the specimen.
How can spinach neutralize the explosive trinitrotoluene, TNT?
TNT is a dangerous explosive. There are more than 500,000 tons of TNT stockpiled throughout the United States. Spinach contains a powerful enzyme called nitroreductase that is able to neutralize TNT by converting it to other compounds that are less dangerous. Through additional reactions, these less harmful compounds can be converted to carbon dioxide gas. This enzymatic process is of great interest to the United States military.
What are the major achievements of biotechnology in the 20th century?
Date Discovery 1968 Stanley Cohen, 1922, uses plasmids to transfer antibiotic resistance to bacterial cells 1970 Herb Boyer, 1936. Discovers that certain bacteria can restrict some bacteriophages by producing enzymes, restriction enzymes. Isolates EcoRI 1972 Paul Berg, 1926, splices together DNA from the SV40 virus NA. Coley, making recombinant DNA, shares 1980 Nobel Prize with Walter Gilbert, 1932, and Fred Sanger. 1918, 1974 Stanley Cohen, 1922, Annie Chang, and Herb Boyer, 1936, splice frog DNA into E. coli, producing the first recombinant organism 1975 DNA sequencing developed by Walter Gilbert. 1932, Alan Maxim, 1952, and Fred Sanger, 1918, 1978 human insulin cloned in E. coli by biotech company Genentech 1986 Kary Mullis, 1944, develops the polymerase chain reaction, PCR, in which DNA polymerase can copy a DNA segment many times in a short period of time. 1989 Human Genome Project. HGP, begins 1990 Researchers at National Institutes of Health, NIH, use gene therapy to treat a human patient. 1994 Introduction of first transgenic food, the Flavor Savor tomato. 1996 Dolly is cloned by Ian Wilmut. 1944, 1997 First human artificial chromosome is developed. 2000 Completion of the first working draft, 90% complete. Of the HGP 2003 Glawfish, the first genetically modified pets are marketed and sold in the U.S.
What feature of liverworts hints to their name?
Liverworts were named during the Middle Ages, when herbalists followed the theoretical approach known as the doctrine of signatures. The core philosophy of this perspective was that if a plant part resembled a part of the human body, it would be useful in treating ailments of that organ or part. The thallus of thalloid liverworts resembles a lobed liver. Therefore, in line with the philosophy presented by the doctrine, the plant was used to treat liver ailments. The word liver was combined with word, which means herb, to form the name liverwort.
How can bacteria survive inside our gut?
Bacteria known as intestinal flora survive in both the small and large intestine because of the neutral pH of the intestinal environment. Because these bacteria have low requirements for oxygen and sunlight exposure, they are well suited to life in this environment. They use our digested food as a source of nutrition and even provide a benefit to us. By synthesizing several of the vitamins, biotin, as well as vitamins K and B5, we need. If the flora are thriving, it is more difficult for disease-causing microbes to establish themselves in the intestines and attack our bodies.
What is the difference between inducible and repressible operons?
Inducible operons are used to produce proteins only under specific conditions. Most bacteria use glucose as their main energy source. Therefore, the unusual arrival of the milk sugar lactose in the cell turns the operator switch to the on position, which allows transcription to occur. Repressible operons have operators that are always on, transcription occurring. Unless a signal, for example, too much product, turns them off.
What is the structure of a virus?
Viruses consist of strands of the genetic material nucleic acid, the basis of a genome, which is surrounded by a protein coat called a capsid. The capsid protects the genome and gives the virus its shape. Viruses may be either helical or icosahedral. Some viruses display a combination of helical and icosahedral symmetry, known as complex symmetry. The capsid is often subdivided into individual protein subunits called capsomers. The organization of the capsomers yields the symmetry of the virus. Animal viruses often form an envelope around the capsid. This envelope is rich in proteins, lipids, and glycoprotein molecules.
How does DNA correct its own errors?
Spontaneous damage to DNA occurs at a rate of 1 event per billion nucleotide pairs per minute. Assuming this rate in a human cell, there are 10,000 different sites in the body at which DNA is damaged every 24 hours. DNA has a number of quality control mechanisms. DNA polymerase, the enzyme that catalyzes DNA replication, has a proofreading function that immediately corrects 99% of these errors during replication. Those errors that pass through are corrected by a mismatch repair system. When a mismatch base, such as Ag, is detected, the incorrect strand is cut and the mismatch is removed. The gap is then filled in with the correct base, and the DNA is resealed.
How can genes be silenced?
Genes can be silenced either by preventing their transcription or by interference with their transcript. Gene silencing is a method by which researchers can study gene expression.
How do plant cells use carbon dioxide?
Plant cells reduce carbon dioxide to sugar by using the electrons that are produced when chlorophyll absorbs light. 6 carbon dioxide, CO2, molecules, along with 6 water H2O, molecules, can be converted into a simple sugar, C6H12O6.
How are the results of paper chromatography deciphered?
Substances that have color are easily seen on a chromatogram. A completed chromatogram is sprayed with ninhydrin and may be heated to detect amino acids. Amino acids turn pink or purple when sprayed with ninhydrin. Molecules of reducing sugars become gray-black when treated with aniline phthalate. Many organic substances become brown spots on a yellow background when exposed to iodine vapors. The distance of the various spots from the origin is measured and used to quantify the movement of the sample. Under standardized conditions, a substance moves at a characteristic rate in a particular solvent system. A researcher can calculate the distance the solvent travels with the distance the substance travels. The result is known as the RF value. Consulting tables of RF values for various substances in various solvents permits preliminary identification of the substance.
Where do scleriids occur in plants?
Scleriids occur in the roots, stems, and seed coats of plants. Groups of scleriids in the soft flesh give pears their characteristic gritty texture. Scleriids also give nutshells and seed coats their hardness.
What does genetics have to do with behavior?
Some ethologists feel that all behavior is genetically programmed. If a behavior is under genetic control, then it is a sequence of events. Sign stimulus, releasing mechanism, and fixed action pattern. However, the mainstream view is that most or all behavior is a result of a combination of genetic programming and environmental learning.
How is yeast utilized in food and beverage manufacturing?
Yeast is used in winemaking, beer making, and bread making. Yeast converts food into alcohol and carbon dioxide, CO2, during fermentation. In the manufacture of wine and beer, the yeast's manufacture of alcohol is a desired and necessary component of the final product. The CO2 is what gives beer and champagne their bubbly effect. Bread making requires the CO2 produced by yeast for certain doughs to rise. Yeasts used in brewing and baking are cultivated strains carefully kept to prevent contamination.
What is DNA supercoiling?
When DNA is not being replicated or specific genes are not transcribed. Its normal form is two strands twisted around a helical axis, much like a spiral staircase turning clockwise. However, during DNA replication or transcription, enzymes alter the structure of DNA such that additional twists are added. Positive supercoiling, or subtracted, negative supercoiling. Either type of supercoiling makes DNA even more compact. A group of enzymes, the topoisomerases, are able to disentangle DNA strands. They are called topoisomerases because they control the topology of DNA.
Why are horsetails called scouring rushes?
The epidermal tissue of horsetails contains abrasive particles of silica. Scouring rushes were used by Native Americans to polish bows and arrows. Early North American settlers, who cleaned their pots and pans along stream banks, used horsetails found in abundance in such areas to scrub out their dishes.
What is biopiracy?
Biopiracy refers to the development of pharmaceutical products without compensation to the native communities that protected and nurtured the organisms on which these products are based.
What is the difference between regular acidophilus milk and sweet acidophilus milk?
Both acidophilus milk and sweet acidophilus milk are inoculated with the bacterium Lactobacillus acidophilus. Many health practitioners believe that this bacterium, a normal member of the human intestinal flora, aids in digestion. Regular acidophilus milk is produced by adding the Bacterium to vats of skim milk as part of the fermentation process. It has a characteristic sour taste. When the bacterium is added to pasteurized milk and packaged without fermentation, sweet acidophilus milk, which lacks the characteristic sour taste, is produced.
What is a niche?
A niche is similar to the job description of a population within a given species. However, instead of describing how many hours individuals will work, it describes features such as whether the species is active at night or during the day, what it eats, where it lives, and other aspects of day-to-day life. From one environment, or community, to another. The niche may vary depending on how much competition the species faces.
How is DNA unzipped?
DNA is unzipped during its replication process, the two strands of the double helix are separated and a new complementary DNA strand is synthesized from the parent strands. Also, during DNA transcription, one DNA strand, known as the template strand, is transcribed, copied, into an mRNA strand. In order for the two strands of DNA to separate, the hydrogen bonds between the nitrogen bases must be broken. DNA helicase breaks the bonds. However, the enzyme does not actually unwind the DNA. There are special proteins that first separate the DNA strands at a specific site on the chromosome. These are called initiator proteins.
How long have leeches been used for medicinal purposes?
Leeches have been used in the practice of medicine since ancient times. During the 1800s, leeches were widely used for bloodletting because of the mistaken idea that body disorders and fevers were caused by an excess of blood. Leech collecting and culture were practiced on a commercial scale during this time. William Wordsworth's 1770 to 1850 poem The Leech Gatherer was based on this use of leeches.
Whose experiments showed that non-lethal bacteria could be transformed into lethal bacteria?
In 1928, an Army medical officer, Frederick Griffith, 1878 to 1939, was trying to find a vaccine against Streptococcus pneumoniae. In the course of his work, he found that there were two strains of the bacteria, one that had a smooth coat S but was lethal. The other form had a rough coat R, which was non-lethal when injected into mice. He decided to investigate what would happen if he injected both heat-killed S bacteria and live R bacteria into mice. To his surprise, the mice injected with this combination died. Upon closer examination of the blood, living S bacteria were found. Something had occurred that transformed the non-lethal R bacteria into S bacteria. Subsequent experiments throughout the 1940s attempted to find the identity of the transforming factor, which was eventually found to be DNA.
How are cell structures divided during meiosis?
Meiosis is just part of the larger process of gametogenic production that occurs in either the ovaries or testes. During sperm formation, spermatogenesis, the cells that eventually become mature sperm go through two successive meiotic divisions. This results in 4 haploid spermatids that will develop into mature sperm cells. The sperm are then reconfigured into a cell that is specialized for one thing, fertilizing an egg. The mature sperm is basically a nucleus with one set of chromosomes, mitochondria, and flagellum for propulsion. In humans, the cells that will develop into mature eggs are present in the ovary before 60. The oogenesis, egg formation, ever occurs. Immature eggs, oogonia, remain in a stage referred to as meiosis I until they mature during puberty. During the stage of meiosis II, oocytes are ready to be released, but will not fully complete the meiotic process until after fertilization takes place. During the progression from one diploid oocyte to 4 haploid cells, cytoplasm is shunted unequally to only one cell. The end result is one large mature ovum and 2 or 3 very small haploid cells called polar bodies.
What are the major types of bonds?
There are 3 major types of chemical bonds: covalent, ionic, and hydrogen. The form of bond that is established is determined by a specific arrangement between the electrons. Ionic bonds are formed when electrons are exchanged between 2 atoms, and the resulting bond is relatively weak. An ionic bond is found in salt. Covalent bonds occur when electrons are shared between atoms. This form of bond is strongest and is found in both energy-rich molecules and molecules essential to life. Hydrogen bonds are temporary, but they are important because they are crucial to the shape of a particular protein and have the ability to be rapidly formed and reformed, as in the case of muscle contraction. The following chart explains the types of bonds and their characteristics.
What are the main organs of the digestive system and their function?
The digestive system includes the mouth, alimentary canal, or gastrovascular cavity, esophagus, stomach, small intestine, large intestine, and anus. The mouth is the opening through which food is ingested. In animals with a second opening for elimination, the digestive system contains an alimentary canal, a tube allowing for the passage of food from the mouth to the anus. In contrast, animals with only one opening have a gastrovascular cavity that serves as the site of digestive activities. The esophagus is another channel through which food passes on the way to the stomach. The stomach, or crop in certain species, such as birds, stores food and is the primary site of chemical digestion. Following digestion in the stomach where food is broken down with acid and enzymes. The food passes into the small intestine. Nutrients are absorbed via the intestine. Much shorter than the small intestine, though greater in diameter, is the large intestine, also called the colon. Here, the solid material remaining after digestion is compacted and then eliminated via the anus.
Do animals other than humans have fingerprints?
It is known that gorillas and other primates have fingerprints. Of special interest, however, is that our closest relative, the chimpanzee, does not. Koala bears also have fingerprints. Researchers in Australia have determined that the fingerprints of koala bears closely resemble those of human fingerprints in size, shape, and pattern.
How did the increased level of radioactive dust, cesium-137, in lichens affect the food chain following the Chernobyl nuclear power station disaster?
Lichens are a primary source of food for reindeer. Reindeer is commonly consumed by humans that live in regions of tundra. When the accumulated level of radioactive dust present in lichens became so high in the reindeer that fed off of them, the reindeer meat became unsuitable for human consumption. Hundreds of tons of reindeer carcasses were disposed of as toxic waste.
How do cells eat?
Cells eat by a process known as phagocytosis, which is important to two types of cells. Amoebas, unicellular protozoans, and mammalian white blood cells, macrophages, neutrophils. Phagocytosis is very important because it provides food for the protozoan. It also plays a critical role in defense systems by eliminating microorganisms or damaged cells in mammals. Once a particle, or microorganism, is ingested, it is wrapped within a vesicle, and then the vesicle fuses with the lysosome. The digestive enzymes of the lysosome then digest the contents of the vesicles.
Who is considered the father of modern zoology?
Konrad Gessner, 1516 to 1565, a Swiss naturalist, is often credited as the father of modern zoology. Based on his 3-volume Historia Animalium, which served as a standard reference work throughout Europe in the 16th and 17th centuries.
What was the role of Maurice Wilkins in early DNA research?
Maurice Wilkins, 1916, was trained as a physicist and worked briefly on the Manhattan Project, where he became disillusioned and then turned to the field of biophysics. He worked at King's College, London, with John Randall, where together they began to use X-ray crystallography to study DNA. Both Wilkins and Rosalind Franklin worked in the same laboratory, but their relationship was not one of cooperation. This ultimately slowed the progress of their work.
In what ways are leaves economically important?
Leaves are used for food and beverages, dyes and fibers, and medicinal and other industrial uses. Certain plants, such as cabbage, Brassica oleracea, lettuce, Lactica sativa, spinach, Spinacia oleracea, and most herbs including parsley, Petroselinum crispum, and thyme, Thymus vulgaris, are grown for their leaves. Berbera leaves, Arctostaphylos uva ursi, contain a natural yellow dye, while henna leaves, Lawsonia inermis, contain a natural red dye. The leaves of palm trees are used to make clothing, brooms, and thatched huts in tropical climates. Aloe leaves, aloe vera, are well known for treating burns and are also used in manufacturing medicated soaps and creams.
How do cells respond to insulin?
Protein-based hormones such as insulin bind to cell surface receptors. While they do not enter the cell, they cause changes in the cell's metabolism. Specific cells in the pancreas secrete insulin, a hormone that regulates the concentration of glucose in the blood. Skeletal muscles and the liver are targets for insulin. Insulin deficiency is responsible for type 1 diabetes. In contrast, type 2 diabetes, also known as adult-onset diabetes mellitus, is not the result of insulin deficiency but is rather due to insulin resistance. Cells with insulin resistance do not respond to increasing insulin levels by transporting glucose into cells.
How does the mark recapture method estimate a wildlife population?
Researchers set traps to capture a population sampling in a given area. The captured animals are marked, or tagged, and then released. After a certain amount of time the traps are set again. The second time, both marked and unmarked animals will be captured. The proportion of marked to unmarked animals gives an estimate of the size of the entire population. The equation used to estimate the number of individuals in the population is marked individuals x total catch second time n equals recaptured marked individuals, where n is the number of individuals in the population. A disadvantage of this method is that it assumes that a marked Individual has the same chance of being trapped as an unmarked individual. Individuals that were trapped once may be wary of traps. Or they may seek traps since they have learned that traps provide food.
What is meant by the modern era of genetics?
Mendel's work was really not appreciated until advances in cytology enabled scientists to better study cells. In 1900, Hugo de Vries, 1848–1935, of Holland, Karl Correns, 1864–1933, of Germany, and Erich von Tschermak, 1871–1962, of Austria examined Mendel's original 1866 paper and repeated the experiments. In the following years chromosomes were discovered as discrete structures within the nucleus of a cell. In 1917 Thomas Hunt Morgan, 1866–1945, a fruit fly geneticist at Columbia University, extended Mendel's findings to the structure and function of chromosomes. This and subsequent findings in the 1950s were the beginning of the modern era of genetics.
What is limnology?
Limnology is the study of freshwater ecosystems, especially lakes, ponds, and streams. These ecosystems are more fragile than marine environments since they are subject to greater extremes in temperature. The study of limnology includes the chemistry, physics, and biology of these bodies of water. F. A. Farrell, 1848–1931, a Swiss professor, has been called the father of limnology.
Do animals marry?
It is estimated that 90% of bird species are monogamous, that is, one male mates with one female to produce offspring. Some of these pair bonds may actually extend beyond a single mating season and so could be considered a form of marriage. The type of pair bonds a species will form is dependent on their ecological niche and is heavily influenced by the needs of their offspring. Altricial offspring, which require large amounts of parental care for survival, like humans, demand the efforts of two parents and therefore are more likely to be found in monogamous species.
What is lyophilization?
Lyophilization is a freeze-drying technique for preservation and storage of bacteria and other microorganisms. Bacteria can be stored for extended periods of time, 3 to 5 years, as frozen cell suspensions or as freeze-dried, lyophilized, cultures. This technique is achieved by placing bacteria in a nutrient broth containing 15 to 25% glycerol and freezing at temperatures of -94 degrees Fahrenheit -70 degrees Celsius or lower. The glycerol reduces ice crystal formation that would cause subsequent cell damage and disrupt biological structures.
What is the oldest known fossil flower?
The fossil of the world's earliest known flower was discovered in 1986. This flowering plant is from the 120-million-year-old Kunwara fossil beds found near Melbourne, Australia. The fossil flower was an important find because all the parts of the flower were attached to an intact plant. The fossil resembles a small black pepper plant, less than 1 in 3 centimeters, long. Paleobotanists believe that this plant represents an ancestral type of flower.
How many acres of wetlands have been lost in the United States?
Since access to water is important to industrial development, many cities are located in areas including wetlands. In the urbanization process, wetlands have been drained, filled, or used as dumps. Each wetland area serves as a habitat to many different plants and animals. With special regard to spawning and nursery habitats. The Wetlands Restoration Act, H.R. 1474, enacted November 29, 1990. Refers to wetland mitigation banking and provides that any person who discharges dredged or fill material into the waters of the United States must have a permit from the Army Corps of Engineers. This act is an attempt to preserve the complex communities that are found within wetlands. Wetlands are the lands between aquatic and terrestrial areas, such as bogs, marshes, swamps, and coastal waters. Although wetlands were at one time considered wastelands, scientists now recognize the importance of wetlands to improve water quality, stabilize water levels, prevent flooding, regulate erosion, and sustain a variety of organisms. The United States has lost approximately 100 million acres of wetland areas between colonial times and the 1970s. The 1993 Wetlands Plan established a goal of reversing the trend of 100,000 acres of wetland loss to 100,000 acres of wetland recovery.
How many different species of protists are known?
Biologists estimate there may be as many as 200,000 species of protists.
Is the color of bacteria of significance?
The pigments that occur in bacteria are helpful in the identification of bacteria. Bacteria can be red, purple, green, or yellow. Some bacteria produce pigments only under certain environmental conditions, such as particular temperatures.
What are the 4 levels of structural organization in animals?
Every animal has 4 levels of hierarchical organization: cell, tissue, organ, and organ system. Each level in the hierarchy is of increasing complexity, and all organ systems work together to form an organism.
What is dietary fiber?
Dietary fiber is a type of carbohydrate that cannot be broken down by digestive enzymes. Because of this, the fiber passes through the digestive tract more quickly, aiding in elimination. The term dietary fiber includes the cellulose found in plant cell walls and the chitin that makes up the support tissues of fungi, mushrooms, crustaceans, and insects.
How many quills does a porcupine have?
For its defensive weapon, the average North American porcupine has about 30,000 quills or specialized hairs. Comparable in hardness and flexibility to slivers of celluloid and so sharply pointed they can penetrate any hide. The quills that do the most damage are the short ones that stud the porcupine's muscular tail. With a few lashes, the porcupine can send a rain of quills that have tiny scale-like barbs into the skin of its adversary. The quills work their way inward because of their barbs and the involuntary muscular action of the victim. Sometimes the quills can work themselves out, but other times the quills pierce vital organs, and the victim dies. Slow-footed and stocky, porcupines spend much of their time in the trees, using their formidable incisors to strip off bark and foliage for their food, and supplement their diets with fruits and grasses. Porcupines have a ravenous appetite for salt, as herbivores. Plant-eating animals, their diets have insufficient salt. So natural salt licks, animal bones left by carnivores, meat-eating animals. Yellow pond lilies, and other items having a high salt content, including paints, plywood adhesives, and human clothing that bears traces of sweat, have a strong appeal to porcupines.
Why do birds flock?
The old adage about safety in numbers certainly holds true for flocking birds and other social groups. Flocks, herds, schools, and others provide a refuge for the young and an anti-predator advantage for all. The simultaneous movements of a large number of prey are apt to confuse predators and decrease the likelihood that they will capture any one individual.
Who was Konrad Lorenz?
Konrad Lorenz, 1903 to 1989, became famous for his work in the field of avian ethology, particularly in his studies of imprinting. By raising goslings from the time they were hatched, Lorenz was able to make the goslings follow him rather than their own mother. This work led to the theory that the goslings were genetically programmed to exhibit a certain behavior with regard to any large organism that was near them during a critical early period of their life. In his publications he applied comparative methods to the study of behavior and the psychology of perception.
What are the functions of the circulatory system?
The primary function of the circulatory system is to transport oxygen and nutrients to all the cells of an organism. The circulatory system also transports wastes from the cells to waste disposal organs, such as the lungs for carbon dioxide and the kidneys for other metabolic wastes. In addition, the circulatory system plays a vital role in maintaining homeostasis.